Suancai, also called Chinese sauerkraut, is a traditional Chinese pickled Chinese cabbage. Suancai was fermented based on microorganisms naturally present on the raw materials. During the fermentation, more and more lactic acid was formed. This would lower the pH value as the graph above. The alternation of the cabbage texture is brought about by the breakdown of complex carbohydrates and proteins, facilitated by enzymes and organic acids produced during the fermentation process. The hardness of Chinese cabbage was decreased with fermentation time prolonged as the graph above. Preparing suancai for an entire winter typically requires a large jar. Fresh cabbage is sun-dried for 3 days to remove extra water, preventing spoilage during fermentation. Subsequently, outer leaves are removed, and the cabbage is cut to fit into the container. Layering cabbage and salt in the jar, the process is repeated until the jar is full. Salt plays a crucial role in shaping the sensory attribute and preserving the product by directly influencing the brine's salty flavor and microbial structure. Moreover, the salt concentration is pivotal in the initial stages of the fermentation process. An optimal salt concentration effectively hinders the reproduction of E. coli and fungi. Diverse salt concentrations result in varied osmotic pressure, water activities, and substance exchange rate. Studies indicate that a 2% salt addition accelerates the maturation of suancai, but does not inhibit undesirable microbial growth. In contrast, a 5% salt concentration can make E. coli and fungi disappear faster. However, an 8% salt addition, while delaying maturation and reducing sugar utilization, does not achieve a quick elimination of fungi and E. coli, making it less desirable. Therefore, the 5% salt concentration leads to a product of finest quality. Compression is essential to minimize air inside the jar. Using picked out leaves to seal the jar ensures no gaps at the mouth. Choosing a suitably sized heavy stone, smaller than the jar opening, as the cabbage diminished in size, the stone descends, preventing cabbages to float up. After 48 hours of fermentation, adding salt water to cover the cabbage ensures an airtight environment. A plastic bag on the top prevents air and dust contamination. After patiently waiting for a month, suancai is ready to be enjoyed. Next, let's systematically review how microorganisms within the Chinese cabbage create this fermentation miracle over the course of a month. Initially, naturally occurring microflora is presented on the cabbage leaves. The heterofermentative lactic acid bacterium, Leuconostoc mesenteroides, takes the lead in the early stages of lactic acid fermentation. Noteworthy for its tolerance to high salt and sugar concentrations, it produces lactic acid, carbon dioxide, and ethanol. The resulting carbon dioxide and acid swiftly decrease the pH, curbing the growth of spoilage microorganisms. The generated carbon dioxide replaces oxygen, establishing an anaerobic environment conducive to the growth of subsequent lactobacillus species. As the fermentation progresses into the mid-stage, Lactobacillus brevis assume dominance. This microaerophilic lactic acid bacterium follows an obligately heterofermentative pathway, employing phosphoketolysis to produce a mix of lactic acid, ethanol, acetic acid, and carbon dioxide during hexose fermentation. Notably, it lacks phosphotransferase system specific to glucose, fructose, and lactose. In the final stage of fermentation, acetic acid bacterium takes center stage. As gram-negative bacteria deposits the capability to oxidize ethanol into acetic acid, marking the conclusive phase of the fermentation process.
Now it's the end of suancai fermentation. Hope this video helps you understand it better, and we look forward to your own suancai. Thanks for watching.